Pharmapal.
Identification of protein interaction partners from shape complementarity molecular cross-docking. Hello everyone! Welcome back to our channel. Today, we're diving deep into the fascinating world of protein-protein interactions, or PPIs, and how we can predict these interactions using advanced computational methods. In this video, we'll explore key concepts, methodologies, and findings from recent studies, particularly focusing on the work by Lane and Carbone. So, let's get started. What are protein-protein interactions? Protein-protein interactions are crucial for virtually every biological process. They regulate signaling pathways and are essential for cellular functions. Understanding these interactions can help us develop new therapies for various diseases. The challenge of predicting PPI. Predicting which proteins interact is a complex task. Traditional methods often struggle to distinguish between true interacting partners and non-interactors. This is where computational methods come into play, particularly molecular docking. Molecular docking explained. Molecular docking involves simulating how two proteins might bind together. The goal is to predict the native conformation of protein complexes. Key terms. Shape complementarity. This refers to how well the shapes of two proteins fit together. Docking score, a numerical value that estimates how well two proteins can interact based on their shapes. Recent findings on docking methods. A significant study conducted complete cross-docking on a dataset of 46 enzyme inhibitor proteins. Here's what they found. Docking score distributions. The study revealed that the docking scores for known interacting partners were not significantly distinguishable from those of non-interacting pairs. Importance of experimental interfaces. By incorporating knowledge of experimental interfaces, researchers could retrieve true interacting partners with high accuracy. The interaction index. To quantify the likelihood of interaction between protein pairs, the researchers developed an interaction index, 2. This index considers FIR, fraction of interface residues, the fraction of the docking interface that corresponds to experimentally identified interfaces. Docking score, the score from the docking simulation. The formula looks like this. IIP1, P2 equals min, FIRP1, P2 asterisk EP1, P2. This approach significantly improved the prediction accuracy, achieving an area under the curve AUC of 0.81, indicating a strong predictive capability. The role of interface subregions. The study further categorized the interface into 3 subregions. Support, residues buried in both conformations. Core, exposed residues that become buried upon binding. Rim, residues exposed in both states. Interestingly, the core and support regions provided more relevant information for identifying true partners than the rim. Positive predictive value versus sensitivity. An essential takeaway from the study is the distinction between positive predictive value and sensitivity. The finding suggests that it's more critical for the docking interface to match the experimental interface rather than merely covering a large portion of it. Main takeaway on cross-docking in protein-protein interactions. Cross-docking is a computational technique used to predict protein-protein interactions, PPIs, by evaluating how well two proteins can dock together based on their shapes and structural features. The study by Lane and Carbone highlights several critical insights regarding the effectiveness of cross-docking, particularly when combined with experimental knowledge of protein interfaces. Key findings. Docking score distributions. The distributions of docking scores for known interacting complexes were not significantly distinguishable from those of non-interacting pairs. This indicates that relying solely on docking scores may not effectively differentiate true interactors from non-interactors. Importance of experimental interfaces. Incorporating knowledge of experimental interfaces significantly improved the identification of true interaction partners. The study demonstrated that the docking conformations for known complexes closely resemble the experimental interfaces, while those for non-interacting pairs do not. Subregion contributions. The analysis revealed that specific structural subregions of the interface, support and core, are more informative than others, RIM, for predicting true interacting partners. This suggests that predictive strategies should focus on these critical regions. Positive predictive value versus sensitivity. The study emphasized the importance of maximizing positive predictive value, the proportion of true positives among predicted positives, over sensitivity, the ability to identify all true positives. This approach is crucial for enhancing the reliability of predictions in PPI studies. Methodological recommendations. The authors suggest developing methodologies that account for confidence levels in potential interacting partners rather than only identifying the best match. This could involve integrating molecular dynamics simulations with ensemble-based docking approaches. Conclusion. The research underscores the potential of cross-docking combined with experimental interface knowledge to identify protein interaction partners accurately. It also highlights the need for refined methodologies that leverage structural insights to improve predictive accuracy in protein-protein interaction studies. Implications for future research. Future studies should focus on enhancing docking algorithms to utilize interface knowledge better, exploring the applicability of these findings across various protein classes. Developing confidence metrics for predicting interactions could be beneficial in experimental contexts where interface data is lacking. Conclusion. In summary, the work by Lane and Carbone highlights the power of combining molecular docking with experimental interface knowledge to predict protein-protein interactions accurately. Thank you for watching. If you found this video informative, please like, share, and subscribe for more content on molecular biology and bioinformatics. See you next time.
Pharmapal.